Welcome to the learning module for DNA structure and DNA replication. This is Dr. Tafke for the Cellular and Molecular Medicines course. After completing this learning module, students should be able to describe the structure of B-form DNA, explain the functions of the different proteins involved in DNA replication in prokaryotes, describe the role of the DNA polymerases in E. coli replication, explain leading and lagging strand DNA synthesis, explain how DNA polymerases proofread and correct mismatch mistakes, describe why quinolone drugs have antimicrobial activity, compare prokaryotic and eukaryotic DNA replication processes, describe the major functions of the primary eukaryotic DNA polymerases, explain why rapidly dividing eukaryotic cells need telomerase activity, explain why nucleotide analogs are clinically useful, and provide some examples. We'll begin with a brief overview of the structure of DNA. The monomeric units of DNA are nucleotides, and each nucleotide is made up of a heterocyclic nitrogenous base, a sugar, deoxyribose, and a phosphate. DNA contains nitrogenous bases adenine, guanine, cytosine, and thymine, while ribonucleic acid, RNA, contains adenine, guanine, cytosine, and uracil instead of thymine. Polynucleotides consist of nucleosides joined by a 3' to 5' phosphodiester linkage. In DNA, two polynucleotide chains are joined by hydrogen pairing between their bases, adenine with thymine and guanine with cytosine. There are 3 hydrogen bonds between cytosine and guanine, and only 2 hydrogen bonds between adenine and thymine. The 2 DNA strands form a double helix, one chain running in the 5' to 3' direction and the other in the 3' to 5' direction. The base sequence of the 2 strands are complementary. Adenine on one strand is matched or paired with thymine on the other strand, and guanine is paired with cytosine. The form of DNA most prevalent in our cells is B-form DNA, which was first described by Watson and Crick. It is a right-hand helix with the base pairs stacked in the center of the molecule, like the steps in a spiral staircase. The phosphate groups are on the outside of the double helix. 2 acidic groups of each phosphate are involved in phosphodiester bonds. The third is free and dissociates its proton at physiological pH, giving the molecule a net negative charge. B-form DNA is a right-hand helix with about 10 base pairs per turn. This arrangement results in a major and minor groove in the DNA. Other forms of DNA are A-form and Z-form, which will not be discussed in this course. Another way to visualize the major and minor grooves of B-form DNA is to look down from the top of the double-stranded helix rather than the side. Now it becomes much more apparent that the major groove is wider than the minor groove. The important point is that the major groove is wider and contains more groups to form potential interaction with side chain residues of DNA binding proteins. This contributes to the specificity of the interaction with specific sequences in DNA with specific proteins such as transcription factors. This space-filling model demonstrates the interaction between an alpha helical region of a transcription factor with the major groove of a portion of double-strand DNA. The DNA within a nucleus of a eukaryotic cell is organized in chromatin composed of DNA and small basic proteins called histones. Histone proteins are very rich in lysine and arginine residues. This abundance of positively charged amino acids allows for tight electrostatic interaction between the negatively charged phosphate backbone of DNA. An octamer of histones comprised of histone H2A, H2B, H3, and H4 comprise a nucleosome core around which the DNA is wrapped. The DNA between nucleosomes is referred to as linker DNA and is associated with a histone called H1. Nucleosomes are packaged into higher order structures, sometimes called solenoids, which are further packaged into higher order structures called nucleofilaments. The nucleofilaments then are associated with nuclear scaffolding proteins to form chromosomes. The chromosomes of prokaryotic organisms are closed circular double-stranded DNA molecules. Replication of the chromosome begins at one specific site called the origin of replication. The origin of replication as a sequence of bases that is recognized by the DNA proteins, which initiate strand separation and the formation of a replication bubble. DNA helicases unwind the DNA in both directions, forming two replication forks. DNA synthesis proceeds in both directions, so it is referred to as bidirectional. When replication is completed, there are two closed circular chromosomes, each with one strand of parental DNA and one strand of newly synthesized DNA. Therefore, the mechanism is semi-conservative. Both strands serve as templates for DNA synthesis, and each DNA molecule that is generated contains one strand of original DNA and one strand of new DNA. Initiation of DNA replication in prokaryotes involves several proteins, including DNA, which recognizes the origin of replication to initiate the events, helicase, which unwinds double-strand DNA to form the replication fork, DNA Gyrase, which generates negative supercoiling to prevent overwinding from the activity of the helicase, single-strand binding protein that stabilizes the unwound single-stranded DNA, and primase, an RNA polymerase that generates primers necessary for DNA polymerase to synthesize DNA from the template strands. Using ATP, DNA helicases at each replication fork unwind the double-strand DNA. The unwound DNA strands are then bound by single-strand DNA binding proteins to keep them separated. DNA polymerases have an absolute requirement for a free 3' hydroxyl group in order to synthesize DNA. For DNA replication purposes, an RNA polymerase called primase, which does not require a free 3' hydroxyl group to synthesize a polynucleotide, synthesizes a short RNA primer. DNA polymerase then utilizes the free 3' hydroxyl group of the RNA primer to initiate DNA synthesis. DNA polymerases read the template strand in a 3' to 5' direction and synthesize new DNA in a 5' to 3' direction. This means that one strand can be synthesized continuously. This strand is called the leading strand. The other strand must be synthesized discontinuously as the template DNA is exposed. This results in the generation of RNA-DNA extension products called Okazaki fragments. DNA polymerase III carries out the bulk of DNA synthesis in prokaryotes like E. coli. Once it combines with DNA and starts polymerization, it does not come off until it's finished. DNA polymerase III can synthesize up to 2,000 nucleotides per second. It is also very accurate, making a mistake once every 1 million nucleotides synthesized. Two other DNA polymerases have major roles in prokaryotic organisms. DNA polymerase II functions in DNA repair mechanisms, while DNA polymerase I fills the gap after removal of the RNA primer during DNA replication. Strand separation by DNA helicases causes a problem. It introduces positive supercoiling in the double-stranded DNA in front of the replication fork. This causes torsional stress in the DNA molecule, preventing DNA replication. The positive supercoiling is relieved by the action of DNA Gyrase, a type 2 topoisomerase. Type 2 topoisomerases introduce negative supercoiling into double-strand DNA by nicking both strands of DNA and passing the DNA strands through the nick and then resealing both strands again in an ATP-dependent manner. The activity of DNA gyrase is an absolute requirement for DNA synthesis in E. coli and other prokaryotic organisms. Quinolones, like naldixic acid and ciprofloxacin, interfere with DNA gyrase and prevent the breakage and rejoining of DNA ends during DNA replication. Therefore, quinolones are widely used to treat urinary tract and other infections. DNA polymerase III has two activities. Its primary function is as a DNA polymerase, but it also contains a 3' to 5' exonuclease activity, which allows the enzyme to proofread its work. If a mispaired nucleotide is inserted into the newly synthesized DNA, it will not be able to correctly base pair with the template DNA strand. This is recognized by the DNA polymerase, and it uses its 3' to 5' exonuclease activity to remove the mispaired nucleotide.
DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase III.
The function of DNA polymerase I is to remove the RNA primer one ribonucleotide at a time from the 5' end of the primer using a 5' to 3' exonuclease activity. DNA polymerase I then fills in the resulting gaps by synthesizing DNA from the 3' end of the neighboring Okazaki fragment. Like DNA polymerase III, Pol I also has a 3' to 5' exonuclease activity which it can use to proofread and remove any mispaired nucleotides. The 3' hydroxyl group at the end of one fragment is then ligated to the phosphate group at the 5' end of the next fragment by a DNA ligase enzyme. This seals the nicks between the Okazaki fragments, converting them into a continuous strand of DNA.
DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Replication of DNA in eukaryotic organisms is similar to what happens in prokaryotes. Synthesis occurs in the same 5' to 3' direction, and initiation requires an RNA primer. The primary differences being eukaryotic polymerases are much slower than prokaryotic DNA polymerases, synthesizing only about 50 nucleotides per second. In addition, the genomes of eukaryotic organisms are much larger than prokaryotic organisms. Eukaryotic chromosomes have multiple origins of replications, which allows replication to be completed in a timely fashion. Eukaryotic DNA is also packaged into nucleosomes with histone proteins. This requires a coordinated synthesis of histone proteins with cell division. Eukaryotic DNA polymerases tend to be more complex and more specialized in their interactions. In addition, the chromosomes of eukaryotic organisms are linear, which means they have ends, which require special processing. Eukaryotic DNA replication is very similar to the process used by prokaryotic organisms. There is a helicase, which unwinds double-strand DNA, and a type II topoisomerase activity to relieve the torsional strain caused by the positive supercoiling generated by unwinding of the double-stranded DNA. DNA synthesis is bidirectional, occurring both on the leading and lagging strand at each replication fork. DNA polymerase alpha has a primase subunit which generates RNA primers. DNA polymerase epsilon then takes over for the synthesis of DNA on the leading strand, while DNA synthesis on the lagging strand is carried out by DNA polymerase delta. Both DNA polymerase epsilon and delta have 3' to 5' exonuclease activity that is used to proofread and correct mismatch errors during DNA replication. RNA primers are then removed by endonucleases and the gaps filled by DNA polymerase delta. The remaining nicks in DNA are sealed by a DNA ligase. Eukaryotic organisms also have two other DNA polymerases, DNA polymerase beta, which is involved in DNA repair mechanisms, and DNA polymerase gamma, which replicates the mitochondrial chromosome. The ends of a linear DNA strand cannot be replicated because DNA polymerase only synthesizes DNA in a 5' to 3' direction and requires a primer with a free 3' hydroxyl group. After removal of the RNA primers at the 5' ends of chromosomes, there is no preceding DNA sequence to act as a primer for DNA polymerase. This would lead to shortening of the DNA strands after successive replications. If rapidly dividing cells, like stem cells, did not have a mechanism to prevent this, eventually, after several rounds of replication, genetic information from the ends of chromosomes could be lost to subsequent generations. To overcome this, most rapidly dividing cells express telomerase, a reverse transcriptase, which is an RNA-dependent DNA polymerase that uses an RNA subunit as a template to generate hexanucleotide repeats called telomere repeats to elongate the 3' ends of chromosomes. Thus, telomerase activity generates extra non-coding DNA at the ends of chromosomes that can be replicated indefinitely by the normal DNA synthesis machinery. Most somatic cells do not express telomerase activity and thus have a limited capacity to replicate. Without telomerase activity, division of somatic cells is limited by cell surveillance mechanisms that monitor chromosome integrity and activate mechanisms of cellular senescence, or activate mechanisms of programmed cell death, or apoptosis. Cancerous cells often upregulate the expression of telomerase to become immortalized.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences, from a few dozen to several hundred. This prevents chromosome shortening.
Analogs of nucleosides that lack an appropriate 3' hydroxyl group can prevent DNA synthesis and slow down the division of rapidly growing cells and viruses. These modified nucleosides can be incorporated into a growing polynucleotide strand, but cannot be elongated because they lack an appropriate 3' hydroxyl configuration necessary for the polymerase to incorporate the next nucleotide. Azetothymidine has an azeto group substituted for the 3' hydroxyl of thymidine. It is an inhibitor of the HIV reverse transcriptase, which is not utilized by eukaryotic DNA polymerases, so it has little cytotoxicity. Dideoxynucleotides, such as dideoxyinosine, an analog of deoxyadenosine, are used in cancer chemotherapies because they are chain terminators and slow the replication of rapidly dividing cancer cells. Other nucleoside analogs replace the ribose or deoxyribose sugars with an epimer of ribose, arabinose. Citrobene, or cytosine arabinose, commonly abbreviated ARAC, is a chemotherapy agent, while vidarabine, or adenine arabinose, is utilized as an antiviral agent in the treatment of herpes. Here's a short quiz to determine how well you've mastered DNA structure and DNA replication.